Well, good afternoon, everybody. My name is Leana Sueja, and I'm the Program Director at AgStart. I'm streaming from AgStart's brand new incubator in Woodland, California, just outside of Sacramento. We're delighted that you could join us this afternoon for the National AgTech Forum on Innovation in Produce Safety. Our program will be as follows. We will hear an overview of the issues from our special guest, Dr. Trevor Ceslo. Then our two entrepreneurs will present, and then we will move into question and answer. Unfortunately, Dr. Trevor Suslow got hung up in travel today, so he won't be with us personally, but he was gracious enough to send us a very provocative and lively presentation that will address a lot of the issues in this space. And I want to remind you that we will have virtual networking following the program. If you would like to speak with our guests via Zoom, please follow the link in your email confirmation following the program to join the virtual networking. And to post your questions, please use the chat function. Well, before we launch in, I'd like to tell you a little bit about the next National AgTech Forum and then a little bit about AgStart. The next forum on August 18th will be hosted by NCBiotech and will focus on cybersecurity in the age of data analytics and what that means for the food supply chain. I hope you can join us. So a little bit about AgStart. AgStart is a startup incubator with a mission to accelerate ag, food, and health innovation in our Western region. We provide startups with education, mentoring, and connections to collaborators, customers, and investors. Last month, we opened a brand new shared use incubator in Woodland for tech innovators in ag, food, and health. We have a 2,800 square foot wet lab with 6-foot mobile lab benches for dedicated use. We also have a certified commercial kitchen for food product innovators. The lab at AgStart is the only combination shared wet lab and commercial kitchen in Northern California. Please visit our website, agstart.org, or feel free to contact me if you would like further information. Well, now it is my pleasure to introduce our two entrepreneurs and our guest moderator. Dr. Nita Neaton is a professor in the departments of food science and technology and bio and ag engineering at UC Davis. His research focuses on improving food quality and safety through innovation like encapsulation, novel and antimicrobials, biosensors, and imaging. Professor Nitin has over 130 peer-reviewed publications and is co-inventor on 15 patents and patent applications. He co-founded Carrisan Technologies, which is based on research from his lab. Dr. Javier Atencia has a Ph.D. in engineering from the University of Navarra in Spain. He's a former research faculty in the Department of Bioengineering at the University of Maryland. Javier is the inventor of microfluidic devices to study complex biological systems. He has published extensively in peer-reviewed journals, including Nature, and has filed over 15 patents. Javier founded pathOtrack in 2014 to commercialize technology that reduces the time for the detection of foodborne pathogens. The underlying technology stems from Javier's work in microfluidics and was awarded Best Invention of the Year in Life Sciences by the University of Maryland. Dr. Trevor Ceslo. He is a Ph.D. in plant pathology from UC Berkeley and an Emeritus Extension Research Specialist at UC Davis in the Department of Plant Sciences. At UC Davis, Trevor has had statewide responsibility for quality and safety of perishable horticultural commodities. His program spanned pre-harvest to post-harvest research and outreach on diverse whole fresh and minimally processed horticultural foods. His past and current research combines lab and on-farm studies in E. coli, Salmonella, and Listeria in both conventional and organic systems. Trevor serves on multiple government and industry panels and advisory boards, including USDA and the Center for Produce Safety. Trevor was the Vice President of Produce Safety at the Produce Marketing Association from 2018 to 2020, and he recently formed Trevor Ceslo Consulting, LLC. And we will now move into Trevor's presentation.
Listening to this recording, the gremlins of travel were unkind to me, and I'm doing the best I can to just provide this as hopefully some points that the panelists participating and the others that are participating in this forum, can use as perhaps springboards for question and answer. What I was asked to talk about was really some of the areas in produce safety, fresh produce, raw agricultural commodities, as well as fresh cut value-added produce that could really present some opportunities for innovation and entrepreneurial activity. And I included services as well as technology opportunities because I honestly believe that there are a number of things, and particularly in this sector, where business opportunities in providing support, and I'll go through a couple of examples, and training in ways differently than what's already out there to actually achieve a higher level of competency and performance, I think are possible. Just thought I'd put up a couple of examples of opportunities that I hear about and people are talking about. But I wanted to emphasize as I go through some of these categories, none of which are truly like novel, but they're sort of what's out there in the conversation right now, and what we see coming through research that I think could be turned into solutions, could be turned into different platforms to help provide supports and implementation in food safety systems. And the point I wanted to make is that I think one of the big areas, can't define the market necessarily, but for scale appropriate and resource appropriate opportunities. So that's really addressing small farm, mid-size producers, shippers, handlers, processors, other sectors of the industry. And I know that you're going to hear about traceability. You'll hear about coating of surfaces and better design for cleaning and sanitation. All of those are absolutely vital and important, but I'm just going to mention a couple of areas really quickly. One is, there's this whole area of digital data. I think the capture and the data management areas are fairly well covered, but really putting them together in a way that makes it easier for this particular sector to participate in that and take advantage of it through analysis and support and analytics. Using that in a way, not that you're going to turn this over to somebody and give them a dashboard and they're going to be able to come up with predictive models within their own data sets themselves. There are a few examples out there, particularly in packing and processing, but really provide that service where you design systems to do all of this. And then you hear quite a bit about these opportunities within digital twins to really take all of this data and then use that to predict risk factors, to identify prioritization in where you're going to spend your food safety dollar. I included novel filtration systems. You may have heard a bit about parasites such as Cyclospora, not particularly susceptible to any of the current antimicrobial chemistries, and some of the things like UV or other types of energetic systems. They're not easily deployed, but coming up with filtration systems that match the throughput and water use to exclude them from surface water sources is something that people are very interested, but also as it applies to water reconditioning to better exclude those risk factors and those targets that are of concern for human and animal health. So wastewater management, reconditioning and reuse of processing water, particularly as we have a global water scarcity issue and a lot of issues around areas where there's drought, and many of the sort of produce packing facilities in rural areas are the biggest consumer of waters and drawing heavily on the groundwater aquifers, and they're being both encouraged but also penalized to some degree to really improve water efficiency and put a lot of effort into water reuse and water conditioning and to use alternative chemistries for water purification to try and get away from chlorine. I'll come back to that very briefly. On the other side, these sort of biological opportunities and really developing systems for sustainable suppressive soils that if a contamination event happens, you're not going to have amplification of at least things like salmonella or E. coli, toxigenic E. coli. And that kind of comes through our deeper understanding of the microbiome, as it is very specific to different regions, different microclimates, different areas, and even different crops, and ways to try and develop ones that can cut across all of that and provide a preventive control. Some of the others, bioaerosols, and ways to identify when during the season, or when you may be in a position to have an issue, by having a cost-effective way to periodically sample and provide an alert to an operator. And then I put this term non-lethal vector disruption. We used to just call it bird harassment, or other ways. There are different technologies that are out there. More could be explored, or better adoption of sonic or other types of things to try and minimize and reduce animal intrusion into production systems. And like I said, in this area of service, there's a lot of training out there. My observation is not all of it is adequately effective in developing the skill sets, developing the operational skills for optimal performance around food safety, and that could be an interesting area, I think. Again, a lot of the emphasis is on these miniaturized sensors for risk exposure alerts. Again, I think there's a lot of discussion around this, and the need for a sort of autonomous monitoring and telemetry to be able to put out an array in the production, because you may have certain risk factors, risk potential. You don't know what the risk exposure is. That may be water, both for crop management, but also in the post-harvest water uses, as I already mentioned, aerosols, but also in monitoring, tracking, what might be going on with biological soil amendments, and monitoring for biomarkers or activities, and linking them together, and ideally cooperatively among regional growers, that they can all take advantage of that data. As well as there are some systems already really monitoring and measuring cleaning and sanitation performance, both for continual improvement, but also for record keeping. I know some of these things may seem somewhat trivial, but they're just sort of thoughts that could be expanded into something hopefully bigger. As I mentioned, I think the key need is in services around data analytics for the sectors that don't have the skills within their employee base, or they're not in a position to tie into a much more expensive system, but for smaller enterprises and really consortia of growers that a business opportunity might develop through that. Chlorine remains the most effective and cost-effective antimicrobial. I suppose you could argue that, but that's sort of, I think, generally held. But there's a very high interest, continued high interest in that remains in novel chemistries. There are many that are effective, but their use is really limited from a compliance basis by the inability to monitor and verify the dose, where you would normally have to do it with a gas chromatograph. But innovating a compact, affordable, operator-friendly, and auditable system to ensure that the validated dose for pathogen lethality is being maintained throughout daily operations. I think a lot of exciting things in identifying, understanding, and then managing or directing the microbiome through different areas. I won't dwell on this a lot, because I'm already taking a little bit too much time. But this area we call niche occupancy, where things have been applied to equipment that's hard to clean, to get it in, to take over space to prevent listeria. It has been and could be more broadly applied to whole facilities, and certainly onto products. Like, sort of hesitating to call it probiotics. That's how it's being discussed, but it fits in.
Leanna, are you able to join again? Nitin, okay.
So Michelle, I believe Liana may be able to join us again.
Hello?
Yes, Liana, you're in.
Yes, hello. So, Dr. Nitin, would you like to start your presentation?
Yeah, I'll start my video.
Thank you.
So I'll share my screen.
Please.
So Liana, can you see my screen?
I can see it perfectly.
Thank you very much. Now, first of all, many thanks to AgStart and the NC AgBiotech for the opportunity to share the exciting technology that we have been working on, and that led to resultant foundation of Carrisan Technologies. The vision that we have for Carrisan is to provide safer, healthier world where critical surfaces are continuously self-sanitizing. So all our vital surfaces in food industry, especially in the fresh produce industry, where there are high risk of cross-contamination, are self-sanitizing in this innovation. So we are all familiar with the challenges of foodborne pathogens. It has a significant societal impact, both economically as well as on individuals who have to go through this painful experience. While the food safety is one of the key challenges, at the same time, we also face significant challenges in the area of food spoilage, where significant quantity of food is spoiled, mostly due to microbial spoilage issues. Or 20% of them is attributed to that factors. And one of the key challenge that is linked to both the foodborne pathogens as well as the spoilage issues is related to cross-contamination by pathogens. So in the current industry, we have many solutions, as Trevor mentioned earlier, which are related to improving sanitation and decontamination of the equipment or the food contact surfaces. But once we clean it, we bring the risk down of any foodborne illnesses or contamination with pathogens. But as the production shift runs through its cycle, and sometimes this can be for a long period of time without significant clean break in between, we increase the risk of cross-contamination. So there's lack of technologies for continuous sanitation during production while the product is being handled. And this is a raw product which is coming from an agricultural setup where it has microbes on its surface. Most of the cases, they are benign microbes. In some cases, there can be pathogens in that environment. Just to illustrate the concept of cross-contamination. So cross-contamination by pathogens can occur as a single batch of produce, which is contaminated, can leave an imprint of microbes on its surface that it comes in contact with it. Now, this surface can become a source of cross-contamination for the clean produce, which did not have the microbes in it to begin with, but can be contaminated from the surface. And this becomes a source of cross-contamination, and this even can continue on a continual basis. So to address that need, our Carrisan Technologies is developing innovative antimicrobial coating, which can be deposited on the surface. And this equipment coating will eliminate the risk of cross-contamination by actively inactivating the microbes that are deposited on the surface, and hence reduce the risk of cross-contamination of the non-contaminated produce. So our solution is to develop an innovative antimicrobial coatings, which make surfaces self-sanitizing. The key innovations in this technology are it kills 50 to 300 times faster than conventional technologies, which is very required because as the throughput of our processing operations is relatively large, so we need to have very fast kill as compared to some of the conventional surface technology, antimicrobial surface technologies. It prevents cross-contamination, reduces the risk of foodborne illnesses. The unique other aspect is it works effectively on legacy equipment. So you don't have to buy a new equipment. You can adapt this on existing surfaces that you have in the facility. It can be applied routinely, and so that it does not build organic matter on it. So it follows a path of your cleaning and sanitation application. So after each sanitation cycle, you can apply or activate this coating on a routine basis. And it'll be sold as a consumable, and it's based on a UC Davis patented technology. And there are many surfaces that our food comes in contact with, and this can start all the way from the harvesting to processing and food service environments. The underlying technology is actually based on bio-based carriers. These are food-grade carriers, which we have combined them with conventional antimicrobials. Now, when we combine this, it provides a very potent antimicrobial composition that has both the affinity to bind microbes in this environment. It also provides very high localized concentration of antimicrobials, and it's made with, as I mentioned, with FDA GRAS materials to start with. And there are multiple applications for the existing technology. And one of the applications we are looking in this case is based on the coating technology that can be deposited on surfaces. The market size is actually relatively large. Now, in the food industry itself, the total addressable market represents both the food plants, the restaurants, the supermarkets, and the convenience stores. In addition to this space, this technology also has applications in healthcare and public health sectors. Why we're talking about developing self-sanitizing, continuously self-sanitizing surfaces is based on the pressures that our food industry has been experiencing, where the food safety issues are of the key interest to the C-level management at this point. And it's based on many factors. There is changes in how we eat, how the food is moving through our supply chain. There's change in the regulatory environment, as well as advances in detection sciences and identification of pathogens. So all of these factors makes us drive our need for better sanitation and more persistent sanitation of critical surfaces. Our business model is simple. It's based on the sale of consumable materials, which we'll be providing to the users who can apply this directly on the surfaces with minimal support. And our go-to-market strategy will be direct sales of product and use of well-established distribution networks. And we have good connections with both the existing players in the retail side, the processing side, as well as the trade organizations and the other segments which are providing services to this industry. And one of the key aspects that I like to highlight is that food safety is considered a pre-competitive issue, which makes working in this direction quite effective, because all these players come together to promote the solutions that are available to the market. Now, compared to other antimicrobial technologies, as I mentioned, that our technology provides a relatively fast kill time. It's usable on legacy equipment. It has persistence of antimicrobial effect after deposition, and can work effectively during operations, as well as the cost effectiveness is highly effective as compared to some of the other antimicrobial technologies that are out there. So in summary, with the unique technology that solves the critical need in our society, relatively large market size, as well as the team that we have put together, we believe that we have a path to success. This is our team here. Myself and Jeff and Zach Bendix are the co-founders, and our roles are there defined for the organizations. We represent significant years of experience in the food industry, both in terms of both the business development roles, scientific roles, as well as some of the leading microbiology research and the bioinformatics roles in this case. We have a panel of advisors who are guiding us in this process as we are developing the technology from the lab scale to the industrial scale applications. And some of the key milestones that we have currently achieved is the awards of the SBIR Phase 1 and grant from the USDA and NSF foundations. And we are building up, starting our lab actually currently right now and beginning our R&D activities and some of the goals that we have for developing our products. With that, I'll stop here and we'll be.
Thank you, Nitin. Javier.
Hello. So let me see how I share my screen. Yeah. This is not it. 1 second. Okay, can you see my screen?
Yes, if you want to put it into presentation mode, that would be great.
I have it at the end. Let me go to the beginning.
Actually, that is
not
your presentation.
No, because you can see here. Let me bring it down. How does it look now? No?
It looks. Okay, now it's just you.
Let me try again. Now?
Perfect.
Okay, well, thank you so much for inviting me to give this presentation and also to participate in the discussion. So basically, I'm Javier Atencia. I'm the founder of pathOtrack, which is a company that is going to revolutionize the logistics and the safety of food. And this is a problem that you all know because of the news and hopefully just because of the news. There's been a lot of outbreaks of E. coli and Salmonella in diverse foods, including meats, also romaine lettuce, and different produce, right? And there's been a lot of outbreaks that has happened also with different, it doesn't matter where you buy your food, you know, it can be, you can buy in Whole Foods or Walmart, the chances are the same and people are getting sick. Some people are dying even, right? So, and it's not just a huge problem from the point of view of public health, but, okay, I cannot move my presentation now. It's not only a problem from the perspective of public health, but it is also a huge problem from the point of view of industry. You remember in 2015, Chipotle had 2 outbreaks in a span of 6 months that cost the company valuation $8 billion. So, and Chipotle's case is just one case out of the 1,000 outbreaks happening in the United States every year, which cost $55 billion, well, in healthcare expenses, but also in recalls and sanitation. So, but what is the problem? So we are in 2021 and we have more outbreaks than ever. and it's not getting better, right? I do believe that the reason why we have outbreaks is because now we have the ability to measure them, right? And connect the dots and realize that actually the outbreaks are happening. But I think that they were always there. Now, one of the major problems with the outbreaks is that if you're in the food industry, if you're a company, you have to produce fast to make a profit because the margins are very small. And to put an example, imagine that you're a grower in Salinas. Well, so that way with time, so time is key, right, to all this, to the outbreaks. And time is expensive. So imagine that you're a grower in Salinas. You know that Salinas in California, it produces 75% of all the leafy greens in the United States. So if you have a farm and you want to sell, for example, to Chipotle, since they had the outbreak, so the Chipotle demands to all the growers to do a test, right, for food safety, to test and to certify that you don't have pathogens in your veggies. So if you want to sell to Chipotle, that means that you have to use a contract lab. You have to take a sample from your field, send it to a contract lab, and they are the ones that are going to run a test that takes 24 hours that will certify that in that sample, there's no bacteria, no E. coli and no salmonella. But what that means is that you cannot ship your product. So you have to hold everything in a refrigerated warehouse, and hold it waiting for the test results. Once you get the positive or the negative, better, right? If it is not contaminated, then you can ship. If you think about this, also the matter of time is that the moment that you harvest, if you do a test, it will take at least 24 hours to get the test results. But you have only 5 days to deliver the product, because if you don't do it on time, you have to toss it. If you want to ship it to the East Coast, it would take 2 to 3 days. And that's why time is so critical. So not everybody does tests. Usually it is driven by the big companies like Chipotle, Walmart, Wegmans, Costco. They demand testing. And because most growers they try to do it as best as possible so that there's no contamination, but you cannot afford testing, for example, post-harvest. It's too expensive, right? We've calculated that for a large farmer in Salinas, if you want to test all your product post-harvest, it would cost you about $5 million to create a new warehouse just to hold the product those 24 hours. Not only that, but we've talked to people, contract labs that are testing for others, and they told us that there are smaller producers that they want to test, but they cannot afford to build a warehouse. So what they do is that they hold the product in the trucks. So we've calculated that a truck loaded with vegetables, just holding the produce, it has to wait 24 hours until you can ship it, which means that it's $50 an hour. Renting a truck is about $1,200 to have the truck sitting, waiting for the test results. And as I said, we've talked to labs, and they told us that that's exactly what happens. They send the trucks to the parking lot of the labs, and they wait there 24 hours. So the big question here is, okay, so if time is so critical, what would happen if one of these contract labs was able to do the same tests instead of 24 hours, 5 hours? And so therefore we can, right? It's pathOtrack. That's what we enable. We are not a lab, but basically it's to get results in 5 hours. It looks 24 hours, 5 hours is not such a big difference. Well, actually it is, because that enables, for example, to be able to stagger production and also warehousing, which means that you need 5 times less space for storage, which is capital spending is reduced by 5 times. Or in the case of trucks, it's very easy to do the math. You would spend only $250 for the truck waiting in the parking lot. But what is it that we do? So we are not a lab, as I said. What we do is that we enable the tests to be much faster. So if you look at the tests that contract labs are doing, they are all accredited by the USDA or the FDA, well, the AOAC, that follows the protocols from the FDA and USDA. And basically the whole detection system in 24 hours consists of 2 steps. The first one is enrichment, which means that you take a sample, 375 grams of lettuce, and you have to add nutrients and incubate for 20 hours, 22 hours. You incubate it so that the bacteria reproduce and grow to levels that can be detectable. And then the detection system usually is just PCR. It's a molecular system that detects the DNA of the pathogen. So this thing called enrichment, has been called the 100 years problem in microbiology, because if the detection systems, there's been a lot of investments and they have improved in the past decade enormously, enrichment has not changed in decades. So that's exactly what we target. Instead of doing the traditional enrichment, we have developed at the University of Maryland and NIST a technology that concentrates the pathogens using a microfiltration system so that they can be detected much earlier. It takes 5 hours to have the detection, the enrichment and the detection. The nice thing of our technology is that it fits in the tests that are used right now in common labs. We don't do the detection, it's just the enrichment. How does it look like? Well, what we are going to do is to sell one instrument one time. That's the one that runs the tests. And we are going to sell disposable devices. Here you have a bag, there's where you put the sample, and it has here the microfiltration unit. So these are disposables, one per test. And the idea is that we will sell one time the instrument, and we will have recurrent revenue from the repeated sales of these disposables. The price of each disposable per test is based on the need of the farmers. And actually, we've done a lot of interviews, and these are the prices that they told us that they would be willing to pay. So our value proposition is that time. This is what we bring. We shorten the time, and time is money, which means for the three different types of people, players in this ecosystem, you have the growers, you have the contract labs that does the tests, and you have the retailers. For the growers, this means that they can save money in warehousing, refrigeration, and they can do more testing. For contract labs, this means that because the tests are shorter, you can do more testing in the same amount of time, or you can reduce the shifts. It looks like the third shift, the night shift, is a conflictive shift, right? So they can just eliminate it. And the third one is the retailers, which means that you can improve the freshness of your food by 1 day. Also, you can, I mean, this is going to eliminate outbreaks, or at least reduce recalls and eliminate outbreaks. So right now in our ecosystem, our competitors would be the big companies that sell detection kits to the contract labs. These companies include Neogen, 3M, bioMérieux, Biorad, and Hygienia. And basically, these companies do both things. They sell kits for enrichment and kits for detection, everything for 24 hours. So we come new into this space where we are going to do is target enrichment kits that are compatible with the existing detection kits from all these multi-billion companies, which means that if they are not only our competitors, but they are our potential partners and potential acquirers. So this was the best innovation of the year at the University of Maryland in 2015. We've raised $1.2 million in a pre-seed round to achieve 2 milestones that we succeeded to achieve. And we have 4 patents. The first one at the University of Maryland. We have exclusive license, and we've developed in-house 3 new patents. So we got funding from the NSF, from NIST, TEDCO. This is an organism in the Maryland state, from Maryland State. And we've participated in the ICOR programs several times, actually. We've done over 500 interviews. And that was before I jumped into the company. I will explain. So as I told you, there are 3 major players in our ecosystems. Contract labs are our first target customers. They conduct about 27 million tests per year. You can do the math. We are going to sell each disposable per test for $40. And we are going to target them first, and corporate labs, it will come later. These are the types of labs that work just exclusively for Tyson Foods or so, and they need high throughput automation. Basically, the target market is huge. This is just for the United States. And our milestones coming up is we applied for accreditation, AgLaunch accreditation. We expect to have it now at the end of the year, and we are going to start sales in produce. So as I said, we are going through AgLaunch accreditation. It's nothing to do with FDA, it's much simpler. And one of the biggest assets of the company is the team, besides the patents, right? We are covering finances, microbiology engineering, and myself. I have a Ph.D. I have over 9 patents. 3 of them were licensed, 1 to Intel. And for none of the other patents I would have done what I've done for this one, which is I quit my job and I embrace the uncertainty of a startup company, because I do believe that we can change the logistics of food. We can have a huge impact in the safety of what we all are eating every day. And I think that this is an amazing opportunity also from the point of view of finances. We have a stellar advisory board that with a couple of serial entrepreneurs, and we are in the middle of a fundraise of $2.5 million to launch our new product for meats, and the new product also a high throughput product for corporate labs. Thank you very much, and I'll be happy to answer any questions and to participate in the discussion now. Thank you.
Thank you very much, Javier. So we have some questions coming in. I may have misspoke earlier. I may have asked you to put them in the chat. That's why we have them coming, both the chat and the Q&A. But if you put a question in the chat, if you wouldn't mind moving it over to the Q&A too, that way we'll have it there and we'll make sure we get to it. So I wanted to start off with a kind of a basic question that came in. I'll read it and then you folks can interpret it for me. It's from Ray Shalito. Some years ago, AOAC did a study on sampling of salad materials from fields and concluded that due to sampling issues, it's not stating what kind, it was a waste of time. Thus, the request for sampling and testing by Chipotle and others is meaningless, is it not?
So the person that wrote the original study, his name is Bob Buchanan, and so he's in my advisory board. Not only that, but when I was working at NIST, I came to him and I asked him, hey, I have this technology. I don't know much about food safety. What do you think? He said, Javier, I think that this is very interesting. I'm going to help you. So that's how I started working, doing research on this. And why is that? It's because it's true that you cannot make your way out of food safety by testing. You have to sanitize, for example, like the other product that Nitin is presenting, right? And you have to put in place a lot of procedures, for safety. But the thing is that that's not enough. You have to measure. If you put in place any procedure to improve safety, you have to have the possibility to measure it. Otherwise, there's no improvement. Not only that, but it's very easy to say, well, if you don't test, it doesn't matter because it's just statistical. Well, California has passed a law now that is banning all the imports of leafy greens from California, from romaine lettuce, unless they do 1 test per acre, usually of 1.5 kilograms of samples, which is a lot of samples. It's crazy because there's no method approved through AOAC to do that. Maximum is 375 grams. That means a lot of money, that will have to be spent on that. And there's one thing, is it needed or not? Well, if you want to sell to Canada, you do have to do that. And Preharvest, the organization, I don't remember the name, LGMA in California, they just passed something similar. So everybody has to test, especially in the regions that are close to feeding facilities, with cattle and animals, which is most of them in the Salinas area.
Well, it's a very important statement that, we have to be safe and sanitary, and we also have to test. So we have an example of both of those here today. So Nitin, talking about sanitizing, we have a couple of questions for you specifically. Is your product specific for bacteria or also against viruses?
Our product works both against bacteria, viruses, and fungal pathogens. There can be some customization of the product to work more specifically towards one, but broadly it works against all 3 categories of pathogens.
And what specific pathogens have been validated with your antimicrobial application? Are there any studies available listing the inclusivity strains of bacteria, virus, and I know that it's listed fungi too. And is it effective against fungi as well?
Yes. So I think that's a good question. So we have published some data actually related to these formulations in peer-reviewed journals, where we have reported the testing against E. coli, Listeria, and Salmonella, as well as some of the fungal strains and some of the viral surrogates, not the human norovirus, but the viral surrogates that are used to validate the inactivation in those systems. And in addition to that, our compositions work very effectively against biofilms also. So they can target the surface bond adherent biofilms, which are more difficult to inactivate than some of the planktonic cells. So works both against the fungal biofilms as well as the bacterial biofilm quite effectively.
Excellent. So going back to the question from Ray, he's noted that it's a sampling issue, not a testing issue. And there is a difference, there is a distinction. And he's saying, I realize that the regulations are in place and we need to test because it's required. Do you have any comment on that?
That's right. At the end, the major driver in produce, it's retailers. And this is driven by social activism, right? But that's a fact. If you want to, we heard about a large farm in Salinas too, that they talked with Wegmans. Wegmans demanded, tests post-harvest, and they couldn't afford it, so they had to let it go. They couldn't take that bid. So, yeah, at the end, that's the reality. Everybody would test if they could. They cannot afford it, and not all the lots are contaminated, so it makes sense. But at the end, money is the major, right? I mean, you want to sell to these big retailers, and that's also, to me, honestly, it's a good thing to know, that Costco, Wegmans, and these places, they demand testing. I mean, we all eat fresh food, and one of my investors in the first round, he has a family office, right? He told me that he invested because he wants this problem to be solved, because he was part of the outbreak, one of the 2 outbreaks of romaine lettuce. He didn't know what he had in New York, and after a month of seeking doctors, they told him that, they said, hey, you won't believe it, but it looks like you have the 0157. It wasn't 0157, it was another stec E. coli. Yeah. And he told me that it was terrible, especially because of the secondary effects, that he has allergies and other problems now. So it's a serious subject, and we have to put all our effort to solve it.
Correct. So Javier, question, is your enrichment technology considered a standard method within international ring test validation, such as AOAC validated?
So yeah, we applied for AOAC validation, but the lab that is going to do the validation, we hired them first to do the same tests, but for us to know that the results that we see in the lab are the same, that are going to come. And basically, we got the confirmation this past Friday with 2 different detection methods that are commonly used in most labs here in US, which means that, do we have AOAC accreditation? No, this is coming. But what it looks like is that, yeah, we meet all the requirements. And I'm very glad that you asked about this, because I've seen many technologies, especially microfluidics, which is my expertise of research, that they claim, yeah, we can detect salmonella in 10 minutes. Yes, but how do you put 300 grams of lettuce inside a microfluidic device? They don't fit. So what you're measuring is not what you have to measure according to regulations, right? There's a lot of innovation, which is great, to replace the detection systems that we have, which work great, honestly, right? But the key to me is exactly that question: is, do you have AOAC accreditation? Yeah. So we are in the process. Short answer.
Nitin, we have a question for you. In terms of your competition, have you compared your technology to other technologies like post-harvest coatings, biological solutions, or cold plasma?
Thank you for the question. Actually, my lab does work in all those 3 areas. So we are putting this as our best foot forward in terms of addressing this challenge of cross-contamination. So for example, in terms of biological controls like bacteriophages and some of the other technologies that we have also done some work in, the typical time to achieve about 2 log inactivation is about on the order of an hour to 2 hours time frame. So, and that is best with the coating. So like the microbes and the phages are right in close contact to each other. Now, that's not acceptable when you're thinking of cross-contamination, because you can imagine that in 1 hour, you can pass significant amount of produce on a surface, especially if your surface is a conveyor belt or a harvesting belt. So it introduces a risk into the system. And once the system risk is introduced, it can propagate further as the product is being processed from washing and packaging and other operations. So now compared to some of the other antimicrobial technologies, like there are technologies related to silver materials, inorganic silver. There are some technologies related to quaternary ammonium salts. Most of them require a close contact.
Oh.
Looks like we have a frozen screen. Let's move on to, well, I'm sure he'll come back. Let's move on to a question for Javier. Let's see. Okay.
Sorry,
question,
I think I got
you are back. Okay.
I'm back there. So I'm not sure where I lost the contact.
Not sure either. Maybe start the last sentence you were on.
So I can restart. So
okay.
I think, so I was giving an example of a biological control, which requires about an hour or 2 hours period to produce inactivation. And similarly, there are other technologies like silver, which requires on the same order of time to produce significant kill, silver or copper, because they're all relying on close contact between the microbe and the surface. Now, this is a big challenge in the food industry because microbes doesn't have to really contact the surface. They can have a conditioning layer of material in between, like the organic content from fresh produce or fat content from a meat processing facility that can form a layer, and the microbes don't have to contact the surface directly. In our case, our technology provides inactivation both by direct contact and also creates an environment around it where microbes are inactivated continuously in its vicinity. So does not require a direct contact necessarily. So that provides a faster kill as compared to some of the other technologies. Compared to plasma and other sanitation technologies, which are upcoming, we have compared our solutions to it, especially when it comes to biofilms. We are way more effective than plasma or other technologies in those cases, especially because those technologies do generate a lot of ROS species, but they need to be targeted to a surface, which is often a challenge in scaling up those technologies. So there are some opportunities there in terms of advancing the solutions with our coatings that is not possible currently with the plasma or other technologies.
So Nitin, I think you may have already addressed this, but there's a question from Lee White regarding the wet environment in much of the produce processing environment and whether that has any impact on the activity of your antimicrobial agent.
No, actually, that's a good question. So we are developing this solution both for wet environment and dry environment. The coating compositions are different for those two cases. Because again, we want the polymer, the coating material to survive the wet handling conditions. So those two conditions does have different dynamics in terms of our composition that we provide a solution for. But our coating does work in both environments.
And then there's another question regarding how often would it need to be applied? In other words, what is the time span that it's effective?
So the current our target is to enable this to be applied after every sanitation cycle. And the coating process we are designing is such that it takes 5 to 7 minutes to apply the coating after each sanitation cycle. And the reason we are going with this approach is because it's very easy to find crevices and cracks on food contact surfaces where pathogens can survive. So if you don't apply regularly coating solutions on it, you can have crack and crevices in your best antimicrobial material there, and you still have pathogens persisting on that surface. So we believe our solution is cost effective, but practically can be applied after every cleaning or sanitation cycles.
Excellent. Okay, a question for Javier. So it's regarding USDA and FDA. How will pathOtrack get past the requirements of the USDA and FDA for the enrichment step. Any regulatory barriers that you've gotten past already? Do you have any inclusivity and exclusivity studies available? And when will it obtain AOAC approval?
Yeah, exactly. So that's a great question. So we are not going to accredit it for all products at first. So we are laser focusing right now on the ones that are most important because of the shelf life, like produce and trim from beef. In trim, it's not just the shelf life, but everybody has got to test because of regulations, right? And we are very specific. We are going through AgLaunch accreditation first with leafy greens. It's like one step at a time, right? I understand. So enrichment, this is a type of enrichment. We have incubation as well. It's just that it's shorter because we can, concentrate the pathogens and we don't have to do so much incubation. That's why it takes such a short time. But we still need to have some incubation. And it's absolutely true. I mean, if you have one single bacteria in the whole sample, you always have to incubate. And anybody that says otherwise, they don't know what they are talking about. It's just statistical. You have to enrich some. But that's part of our process, the 5 hours, right? In terms of inclusivity and exclusivity, our method is not, it doesn't target a specific pathogen. In that, if you test for E. coli, we have developed, tested with several types of E. coli, STEC, and also 015787, and we get the same results. The same with Salmonella. Salmonella is different because the growth rate is different, right? So what we claim right now is Salmonella and STEC, E. coli STEC, which are the ones that they are testing for leafy greens. And for example, Listeria, we've never done tests, mainly because the first person I worked with, the lab technician, she was going to get married. She wanted to get pregnant. I said, well, we are not using Listeria in our lab at all because it's dangerous for the fetus. But anyway, we have to go one by one. So this is our first product. And yeah, AOAC, what it certifies, and the experiments that they did for us in the external lab is exactly that. We get the same results as the FDA BAM protocol.
Very good. So we're approaching the 2 o'clock hour. We can continue the question and answer, and you can speak directly to our guests in the networking session. So if those of you who want to jump over there, you can look in the email you received, confirmation email, and find that link and jump on over there. I'll ask one more question to Javier, and then we'll conclude today's webinar and move over to networking. So Javier, a question about an animal-based food pathogen, Vibrios, or other that are present in seafood, particularly oysters?
We haven't tested it. Is it possible to do? Probably yes. And there's other pathogens also in waters, they have told us, right, for irrigation and all that. The only thing is that based on all the interviews we did through the ICOR program, it's not just having the technology, but having the market, right? We are a small company. We have to get to market. We'll have to raise this second raise. I think that's the last money that we will need until, to make it succeed. But we have to be strategic about that. First, in Salinas, they asked us actually, when we talked to the producers there, if we can set up a lab there. And I told them, hey, we just sell this. It will be inside another lab. Labs are not our business, right? But that's the eagerness that they have right now for our product. And it's not the product really. What they have is to solve their problem, which is time. Time is critical, right? So Vibrio, if there's a need, and I would love to talk more with you about that, if there's a need and the market is there and so on, absolutely. We are open for business.
Well, thank you very much. Thank you, everybody who participated today. I hope we'll see you over in the networking session, but this has been a great webinar. Javier, did you have your finger up?
Yes, one question, Liana. I don't have
okay, sure.
To the networking meeting. Are you going to?
So the networking, Michelle sent that in the invite that she sent to all of us. So if you go back to that email where you had your link,
it's in the chat for everybody else.
You'll find it.
Okay, perfect.
Okay, very good. Well, thank you, everyone. We hope we'll see you in the networking. And this is a very important topic, which will be receiving much more attention in the future. And we thank you again for your participation. Have a great rest of your day.
Thank you very much.
Thank you.